Thank you, everyone, for tuning in at the CRISPR 2.0 conference. And I appreciate you giving me a few minutes to have a chance to kind of go over some of the technology that Fyodor was talking about in terms of work that we're doing at Selecta that we're trying to make available really to as many labs as possible. Selecta is a small technology-focused company, South San Francisco, Mountain View, California. It's about a half an hour south from the city. And it started 14 years ago with seed money from research grants. We're not venture funded. We're very technology focused. And the funds were used really to develop technology for functional genetic screens to identify the genes responsible for disease progression, drug resistance, cell differentiation, et cetera. And really the focus is to use broadly parallel simultaneous type assays to look at large numbers of genes at a time to try and solve the problem really of you know, how are the 20,000 or so protein coding genes plus the other long non-coding and all the other genes in the genome regulating biology. And the platforms try to leverage, the platforms do leverage next generation sequencing heavily, all of our approaches, because with next generation sequencing, with the development over the last 15 years, the NGS readout makes it possible to pull out results from thousands of individual different components of an assay. And so you can really look at 20,000 protein coding genes in a single assay and extract what's going on with each one of the targets that you're looking at. And so we've had some success. Oh, and the idea is really to develop that with the NGS technology in such a way that it doesn't require large-scale automation, LIMS systems, or other industrial infrastructure. We want platforms that really you can use in more or less standard laboratories using standard cell biology and molecular biology approaches. And we've had some success with this approach. Over the years, we have now, I think, on our website, over 300 different publications from various labs that we've either collaborated with or have been customers. And this is just a brief sample of them shown here. But you can go to our website. There is a search function. And if there are particular applications or systems that you're interested in looking at or using, you may be able to find something similar that's already been done. The first technology I want to talk a little bit about is our driver map expression profiling platform. This platform enables you to really get data on which genes are activated, turned on, turned off in different samples, under different treatments, different culture conditions, et cetera. This transcriptome profiling type approach is in some ways similar to RNA sequencing, which is a very common and typical approach. But RNA sequencing, though it's used pretty widely, is very powerful because you're looking at really changes in expression across the entire transcriptome. There are some challenges with preparing the samples for that or running the samples. RNA-seq does require reverse transcribing this whole transcriptome essentially, and then you're doing data analysis of the entire transcriptome after sequencing a fragmented version of that, and then trying to align all of that to the genome. And so you really need to establish a robust pipeline that enables doing that on a regular basis. For expression profiling a small number of genes also, the QRT-PCR approach is really still the gold standard. It provides very precise and reproducible, accurate data. The problem really with QRT-PCR is the multiplex approach. You can really only look at a handful of genes at a time. And so DriverMap attempts to combine the simplicity of the QRT-PCR type of approach, just using a standard RT-PCR reaction, in this case a multiplex PCR reaction, but on a more global genome-wide type of scale. So you can get comprehensive data similar to RNA-seq. It is a targeted approach, so you're looking at a specific set of genes, and in this case, we've scaled it up to be to target all 19,000 human protein coding genes. And so we have specific primers that are designed to target 80 to 200 nucleotides of the transcript of each one of these protein coding genes in human cells and amplify that. And so it's a single multiplex RT-PCR reaction that starts with total RNA. You amplify 19,000 amplicons. And while that may seem like a lot of amplicons to sequence, it certainly is a lot of amplicons to amplify, and it has taken some optimization there. Once you have that, actually that's much less complex than reverse transcription of the whole transcriptome that you get with RNA-seq. And so After you do the amplification, do a second amplification, add the P7 and P5 primers. This is all done in a single tube reaction. Then you can load that onto next generation sequencing. And with 5 million reads of driver map, you can get actually a very comprehensive profile of all of the expressed genes. As compared to with RNA-seq, even with 25 million reads, you can often lose some of the low expressors. And this is because it is a targeted reaction, and you're looking at a much less complex mix of 19,000 amplicons as opposed to the complete reverse transcriptome in a RNA-seq type of approach. Since it is PCR based also, it's very sensitive. You start with total RNA, and you can start with total RNA from down to single cell levels. And we have done this routinely with different samples. And this is just a test experiment where we diluted JERCAT and HEK293 cells in a microtiter plate using a fax down to single cell levels, and then profiled each of these, and we're readily able to tell the difference between the two of them. And so you get sensitivity both in terms of the level of sample, the level of total RNA that you can use down to picogram levels, and looking at low expressed genes. This is also validated by a study done at the beginning of last year, looking at markers for maternal blood in pregnant women to try and predict which women may have trouble with actual labor or delivery of the child. And what they did is they used 3 different techniques to look for markers in maternal blood: driver map, RNA-seq, and also standard QRT-PCR. And they found that the driver map approach, they were able to identify 3 times more protein coding aligned reads than with the driver map approach, with the RNA-seq approach. And the sample-to-sample variance was much better than with RNA-seq, and it correlated better with QRT-PCR, which maybe you would expect because both the QRT-PCR approach and the driver map approach are PCR-based. In addition to the performance advantages, it is actually easier, as I mentioned, because you're starting with total RNA. You don't need to do RNA depletion, ribosomal RNA depletion, globin depletion, isolate, enriched for mRNA. The fragmentation and all of this is done in separate reactions for RNA-seq. With driver map, this is all a single multiplex PCR reaction, and then you go right to sequencing. So you can easily do a plate in a day. And then the next day come in and take the data and align the reference amplicons in the with the actual sequencing data that you're getting, where you have the targeted amplicons and you know what you're looking for. So this can be done very quickly, and it's data that can be shown on a spreadsheet. It's much easier than aligning to the genome as you have to do with RNA-seq. So driver map and other expression profiling techniques provide an understanding of which genes are up and down regulated, but they don't really give you an idea of which ones are required for certain reactions, certain responses in cells. It doesn't tell you which ones are driving the phenotypic changes. And to address that question, it really requires disrupting or perturbing genes to see if they have an effect on the phenotype of interest. If you're looking for development of a disease, for example, or some sort of differentiation, which genes are actually required for that change to occur in the cells that you're looking at. And there are really only 2 technologies that enable this sort of gene disruption analysis on a whole genome level in a single assay. And that's RNAi type of screening or CRISPR type of screening. And that's because with both of these approaches, you can develop a set of reagents that target each one of the targets that you're interested in, and for thousands of genes all across the entire genome, just looking at a database. And so you can make short RNA that target transcripts for shRNA type of screening, or target the genomic region for the genes with CRISPR and disrupt that particular gene, and then see what the change is on the phenotype that you're looking at. And so because of that, CRISPR screening has become a very powerful approach to identify genes responsible for looking at biological changes and seeing which ones are actually causative versus which ones are just changing as a result of the change in the expression. And so the approach there is the DNA encoding the sgRNA is synthesized in a pool and then cloned in a large-scale pool. So it's very easy to clone, for example, 80,000 sgRNA that are targeting all 19,000 protein coding genes with, say, 4 sgRNA each. And then clone these into a lentiviral vector and then package them into a lentivirus and introduce them into a cell population. And you can adjust the virus to cell ratio so that you're getting approximately 1 sgRNA in each cell that you're looking at after selection. And so then and you can have, for example, several hundred cells with each one of these different sgRNA and see what's happening to those subpopulations of cells with a particular sgRNA in your screen. And that's done by allowing the cells then to grow over a period of time and then isolate the genomic DNA and sequence it. The most common application of this is really to look for essential genes. And so in that case, what you're doing is looking for cells where the sgRNA is toxic because it's targeting a gene that's essential, and that results in the cell dying over a period of time, over, say, 10 doublings. And then when you do the isolation then of the genomic DNA and sequence and look for the representation of each of the guides in that population, you'll find that some of the guides have depleted because the cells have dropped out of the population over a period of time. And those are the ones then, such as shown here in the graph where the experimental is graphed against the baseline, that appear below the diagonal that are underrepresented in the experimental population. Those are the ones that are targeting essential genes. And this sort of screen is useful for identifying genetic vulnerabilities, obviously, cancer researchers are interested in identifying these sorts of genes generally, but also if you have particular drugs where certain cells are sensitive and certain cells are resistant, many people are using these type of screens to identify which genes are important for the resistance phenotype to a compound or therapy of some sort. There are also other options too. You can also do screening that's more of a positive screening where you're trying to kill all the cells with some sort of factor such as TGF-beta and then looking for the surviving population, the cells that are resistant to that where the mechanism of action, in this case, say apoptosis, is not effective. You can also have reporters and use FACS type of assay to select cells with activation or deactivation of a particular reporter. There are many different variations that can be used, but as I said, looking at essential genes is really probably the most common. And so, as everyone I'm sure in this presentation is aware, the CRISPR works by knocking out a target where you have the sgRNA localized to a particular region. You have the sgRNA localized, the Cas9 nuclease, to a particular region of the genome, and then introduce a double-strand break, which then, after a while, there's a failure to repair that break correctly, and you get an indel, and that knocks out your target gene. We're interested then in using this system for screening, so we're interested in ways of improving it. And if you look at the bacterial version of the CRISPR system, there are 2 sgRNA molecules, the transactivator or the tracker, and then the guide. And these 2 molecules then hybridize at this hybridization domain and create the binding structure for the Cas9 nuclease. In the bacteria, then this targets a invading virus of the bacteria. So Dune and Charpentier in 2012 created a single guide molecule which combines both of these original RNA from bacteria, and the single guide is shown here by creating a stem loop with this area that this first structure that hybridizes in the bacteria. If you look at that structure, though, there's a couple things to note. One is that it's somewhat shorter than the hybridization domain in the original bacteria. And the other one is that it does have a row of U's, and these row of U's is a weak stop signal for the U6 promoter, which is typically used to express sgRNA mammalian cells. So the Weissman lab at UCSF noticed this and made some modifications to the sgRNA by adding some bases to extend this stem loop structure and presumably then create a better binding site for the Cas9 protein. And then also, if you look at the string of T's used in the RNA, but T's in the genomic DNA, you can maintain the structure, but just simply swap out one of the U's for an A on the opposite strand. And then this then gets rid of that string of T's, the 4 T's, that is a weak stop signal for the sgRNA, for the U6 promoter. And so these two modifications we refer to as the AT inversion, and then also the hybridization domain extension. And so one is the AT inversion, so combination of that is heat, right? And so the question is, there was some work done by the Weissman lab showing that the deactivated Cas9 does bind more strongly to the extended hybridization domain. But the question is, does that give you better knockout actually of your target? And if it does give you better knockout, can you also get better signals when you're actually doing a genomic screen? And so we looked at that by making 4 different libraries, one with the standard wild type sgRNA. By wild type here, we mean the one that Doudna and Charpentier had originally defined. And then the AT inversion, the hybridization extension, and then the combination, the heat structure. And these are 20 different essential genes in cells that have been transduced with these libraries. And there are 8 different sgRNA targeting each of these genes in the library. And so you can see in the results that the standard sgRNA does give you significant depletion for most of them. But when you incorporate either one of these 2 modifications or both of them combined, you're getting significantly higher depletion for the sgRNA that are targeting these genes. And so we routinely then, after this type of work that we were doing, incorporated the heat design into all of our sgRNAs and sgRNA libraries. And it's we see that it gives you better signals than when actually screening. It doesn't just simply increase the binding affinity of the Cas9. So as mentioned, the sgRNA binds to the genomic target sequence and recruits a Cas9 nuclease to induce a double-stranded break in the standard CRISPR type of approach. However, UCSF, they also introduced a couple modifications that deactivate the nuclease function of the Cas9. And so you have what's called the deactivated Cas9, often called the dead Cas9, dCas9. And so it's not completely dead because it still binds to the sgRNA. However, since it doesn't cut now, it provides a platform that you can use to localize this complex to any region of the genomic DNA of interest. And so then you can link other types of activities to that, such as a transactivator, and then be able to localize that activity to a particular region of the genome and modify the genome at that particular location or act on the genome. And the CRISPR-I and CRISPR-A variations of the system have come out of that, where the CRISPR-I inhibits the expression of a particular target by linking a repressor to the dCas9 complex, or transactivator can be linked to the dCas9 complex and upregulate a target of interest. And with, for example, the CRISPR-I approach, you can get a type of screen very similar to a knockout screen where you're getting a depletion of a set of sgRNA based on targeting genes and downregulating those genes that are essential. But unlike the standard CRISPR system, you're not doing a knockout. So the one advantage here is that the system is somewhat reversible, even though it's not quite as robust as the knockout, because the knockout system, I have to say, is pretty clean. And then the reverse, if you're targeting specific genes with the activator complex, you can get upregulation of endogenous genes in a particular system, and that's shown here. And as you can see, it's you're getting some various levels of activation, and some don't activate as well. So as I said, the system is not completely robust and not like knockout, but there's not really another alternative to activate endogenous genes sort of across the genome, just based on the sequence of the the targets that you're interested in, other than this approach. So it's interesting for a kind of a type of screen. And so in terms of ways to improve that, we looked at not just using a single sgRNA, but like most transactivators in cells, you have multiple transactivators targeting a particular promoter region of a gene to upregulate it. And so we tried using, for example, 2 different sgRNA, both expressed in the same cell. And you can see that for the MyoD gene, for here in this example, one of them really gave very small activation, and another one gave a little bit of moderate activation, about 10-fold. But when both of them combined together, we're actually be able to get significantly higher activation. And so we're getting some sort of at least additive effect. And that brings up the idea then of using constructs where you're expressing 2 different sgRNA. And for the CRISPR-A system or CRISPR-I, you can have those targeting the same locus, the same gene, not the same locus though, but the same gene in the promoter region, the upstream region of the gene. And if you have both sgRNA on the same transcript in the same cell, you're getting an additional level of activation than you're getting with either one independently, as shown here. So, you know, we then wanted to see how this also affects, because all of these are done on sort of an individual scale, and much like the heat structure for the sgRNA, we want to see how that works then on a combined scale in a library screen. And just to go over this kind of quickly, it's not so simple then to just make 2 libraries and then new screens with each, because you're really only looking in the library at the phenotype level, and only a small percentage of the genes are actually giving you some effect on that phenotype. And so it's not clear whether there's the single sgRNA or dual sgRNA. It would be difficult to interpret the results. What you really have to look at is at the molecular level, at the expression level. And so to do that, we actually transduced cells with both libraries, fact sorted them, and put about 10 cells in each well, and then used the driver map system to identify which which RNA were changed in the cells in each one of the wells. And then we also extracted, of course, DNA and looked at the sgRNA in each of the wells. And so just to cut a long story short, we did see some difference, and I can go through this experiment in a little bit more detail if anybody has questions. But we did see some difference when we looked at the results. We found that the dual sgRNA library gave, for example, only about a quarter of the genes were not really affected, did not actually increase in activation. Where with the single sgRNA, about a third. So there's some difference there. In terms of the moderate level, about half of the genes were moderately affected, at least about a 10-fold level of increased expression. And then again, the dual sgRNA library increased the expression level of some genes, about a quarter of the genes, 1,000-fold, close to 100 to 1,000-fold, where less were done with the sgRNA. So if you look at this data, you can see that there is some slight skewing, and basically you get a shift of the curve with the dual sgRNA. And this, plus based on the experiments with the individual targeted genes that I showed you, it seems that it's better to have 2 sgRNA targeting each gene than 1. And so we have these libraries also available where they have 2 sgRNAs targeting each gene of interest for CRISPR-I and CRISPR-A. And so with that, I'd just like to wrap up quickly by pointing out that this approach of making libraries is not really limited to sgRNA. Large-scale pooled libraries is not really limited to sgRNA or shRNA for that matter. But you can use it to also make pools of hundreds of thousands or even millions of different unique sequences of barcodes that can be transduced into a cell population, and then uniquely label each cell in that population when you do it at a low MOI, so that you can sorry if that's coming up, so that you can then have a population of labeled cells. And this type of labeling can be useful for a number of applications. For example, you can look at cells, how populations of cells change over time. Hold on one second. How populations of cells change over time with differentiation, implantation, etc. And also maybe whether certain subpopulations of cells have specific characteristics that increase or decrease their ability to survive under specific conditions. This was an example shown here from scientists at MD Anderson, where they used the barcode libraries to label a PDX cell population, and then looked at the effect of various drugs on the subpopulations in that tumor, the cells derived from the tumor. In addition, the barcodes can be combined with sgRNA, and that gives you a way then of tracking sort of the specific changes the sgRNA make on cells in a single cell level. So each cell that picks up an sgRNA also picked up a separate individual barcode. And this study shown here from researchers at Stanford show that how you can quantify tumor development in response to specific knockouts of specific various tumor suppressors in an in vivo mouse model, which is an interesting approach. The similar approaches could be combined also with single cell 10x genomics type of sequencing to look at expression profiling and how particular sgRNA affect the expression of individual populations of cells in a large cell population. So I went over those last couple slides very quickly, but my intention was really just to provide you a quick overview of the technology and the capabilities that we try to offer researchers. And if you're interested in discussing any of these further, these platforms, or you need more information about some products or services, I'd be happy to talk to you about that. And again, I would like to thank you for your attention, and I would be willing to address any questions you might have.
Well, thank you. That's a very, very impressive capability, and it's exciting how much you provide. One's mind starts swarming with experiments. The world has certainly gone a long way since the 90s, when you ordered individual oligos and waited for them. Questions, folks? We have just a little bit of time because of earlier delays. Any questions for Paul? Alice is asking, why do you think you saw the additive effect?
Yeah, that's, I think, as I said, that in normal transactivators, you normally have multiple domains, in a promoter region to get some sort of additive effect in terms of upregulating the gene. In this case, I think there may be some of that. I think in some cases, you have a weak sgRNA and a strong sgRNA, and it's just very much, an additive effect that you're increasing the expression level. But I don't think that there's a general answer across the board. I just think that you increase the chance of actually getting some activation of the target gene by having at least two guides targeting that one from each one of the constructs.
Great.
So.
All right, thank you. There are no other questions. Paul, thank you.
All right, thank you.